Hello. In week 7, we're talking about sequencing projects. This video, I'm going to talk about reduced representation genomic sequencing. Specifically, I'm going to talk about RAD sequencing. To think about why we might want to do reduced representation sequencing, I'm going to walk us through a case study. So in this case study, our goal is to find genomic variants associated with sex in tuna. This is a real problem. Tuna, like a lot of other marine organisms, have no obvious features of sexual dimorphism. That is, I can't look at this fish and say that is a male or that is a female. And again, as always in this class, I'm using the words male and female to describe whether an individual produces small gametes or big gametes. And you can imagine that environmental managers, people who manage populations of this valuable resource of tuna, might be concerned with monitoring the number of males and females in their populations because, of course, these are sexually reproducing organisms, and it would matter if that dynamic changed in your population. So, genomicists are trying to find a genomic way to sex or figure out if the individual fish they're looking at is going to produce small gametes or big gametes. You are the genomicist tasked with answering this question, and here's what you have. You have 100 DNA samples from confirmed female fish. That is, you dissected the gonads and saw if there were small gametes or large gametes present. You also have 100 DNA samples from confirmed male fish. There's a reference genome available for this species, and let's say that you're working with a sequencing facility that charges you $3,000 per lane of sequencing. A lane of sequencing is a unit that the instrument can run at any one time. You can buy multiple lanes if you want to. And this particular sequencing machine returns 300 base pair single end reads, and the throughput of this machine is 400 million reads per lane. And what do you want? You want 20x depth of coverage at every base of the genome for all 200 fish. The first thing we need to figure out is how many reads total do we need? And we can revisit the formula that we use to calculate depth. Here we know that we want 20x depth of coverage. We know that our reads are going to be 300 base pairs. And we know that the genome size, I told you have a reference genome for this species, and I'm telling you now that it's about 800 mega base pairs or 800 million base pairs in length. So now we're going to solve this math to figure out the number of reads that we would need to sequence a single genome at 20x depth of coverage. I'll let you do that math, but if you solve that, you can see that you're going to need about 53 million reads per DNA sample that you have, that is per fish. And when I multiply that by the total number of fish, I see that I need about 10 billion reads total to get 20x depth of coverage for my 200 fish. And now I remember that I can get about 400 million reads per lane. So it's going to take 25 lanes worth of sequencing for me to get those 10 billion reads total. And considering that we're paying $3,000 for one lane of sequencing, if we're going to have 25 lanes of sequencing, that's going to cost us about $75,000 just in sequencing costs. That doesn't even consider DNA extractions and library preparation and all of the power that it's going to take to analyze those data, paying people salaries. So as you can see, this simple experiment is getting really, really expensive. So maybe this isn't the right way to go. Now, I'm sure I over-exaggerated a lot of those numbers, and I definitely did a lot of rounding. So the exact math there is going to be different in a real-world application. But at the end of the day, the point remains that that is a huge sequencing effort for your question. And how do we get around that? Well, we need to remember something really important, and that is that not every base in the genome varies in a species. Here, we are specifically concerned with finding a genomic variant, a sequence variant that varies from between male and female fish. Well, we don't need to look at every single base in the genome because a lot of those bases in this species are going to be identical in every member of the species. So what we want to do is just take a peek at a subset of the genome and see if there are any loci, any variants in the loci in our reduced representation of the genome that covary with sex. So our problem is that we have an 800 million base pair genome and we don't need a marker at every single site. That is, we don't need a very high marker density in order to answer this question. So how do we reduce our marker density? That is, how do we sequence fewer sites in the genome, but still find the variable sites that might be associated with our trait of interest? Well, assuming that these variants are randomly spaced across the 800 million base pair genome, which is of course separated on multiple chromosomes. So pretend that these are chromosomes stitched together when you're looking at this line. One thing that you can imagine doing, as long as you do it randomly enough, is to just chop this genome up into a bunch of little pieces and then just sequence those, hoping that you're going to catch enough variants in those little pieces that you've chopped up. Now the question is, how do you randomly chop up a genome? Evolution has provided us with one solution in that restriction enzymes recognize a specific DNA sequence and they cut the genome at that position. These originally evolved in bacteria to prevent bacteriophage infections in bacterial cells. That's why they're called restriction enzymes, because they restrict the ability of that virus to replicate in bacterial cells. So, what we can do in real life is find a restriction enzyme that recognizes some pattern in the DNA. We can add it to our DNA, and every time that restriction enzyme sees that particular sequence that it recognizes, it cuts the genome. And we hope that these sites, because they're not too long, these sites, sorry, I was going for my laser there, because these DNA sequences aren't too long, we hope that we will encounter those sequences in regular patterns randomly throughout the genome. Another cool thing about using restriction enzymes to chop up the genome is that you can control the sizes that you chop up. That is, you can control the density of markers that you sequence based on the enzyme that you choose. So for example, this is the actual recognition sequence of an enzyme called SBF1, and this is the actual recognition of an enzyme called FAT1. And you can imagine here, refresh your intro probability type skills. Which sequence do you think is going to be represented in the genome more? This 8 base pair long sequence or this 4 base pair long sequence? And if you go through that thought exercise, I think you'll see that one of these enzymes is going to cut up the genome a lot more times than the other. Let's say that you perform a restriction digestion of your 800 million base pair genome with SBF1, which has an 8 base pair recognition site. If every one of the 4 nucleotides are equally represented in the genome, we can expect with some basic probability that we would encounter this 8 base pair recognition site once in every about 65,000 base pairs of sequence. And in your 800 million base pair genome, that means you're going to find it about 12,000 times. So what I've drawn here is, of course, don't count them up. It's not going to be 12,000, but you can imagine that it's going to cut all across the genome. And there are a number of ways to do this. You can actually use more than one restriction enzyme, or you can use one restriction enzyme, but then perform some sort of mechanical shearing to make the bits smaller, or you can use a special kind of restriction enzyme that actually cuts in 2 places instead of once. Regardless of the exact details of how you do this, you're going to perform some side selection and end up with a couple thousand of what we're now going to call RAD tags. RAD tags, where RAD stands for Restriction Site Associated DNA, are the bits that we would actually sequence. The RAD is based on the process of restriction digestion that helped you generate this subsampling of the genome, or this reduced representation, meaning the bits that we're actually seeing in our sequencing only make up a small component of the actual genome. And the tags are related to the fact that there are going to be unique sequences in here that will be representative of the entire genome. Now, since I have reduced my genome to only these 12,000 RAD tags using restriction enzymes, and based on the fact that I don't have to sequence even the entire length of the tag, it depends on what size you selected for. But let's say that I selected for fragments about 1,000 base pairs long, it's still possible for me to just sequence the first 300 base pairs of each tag. So now, based with the materials I said you had access to at the beginning of the problem, you can imagine that you could achieve 20x coverage per RAD tag, that is, to sequence, let's say, RAD tag 1 20 times at this one particular spot for 20x depth of coverage right here and here and here. To do that, you would only need about 240,000 reads per fish. So to achieve 20x coverage for every RAD tag in your 200 fish, you would need about 48 million reads, which, as we said at the beginning of the problem, each lane of sequencing that costs $3,000 gives you 400 million reads. So now, suddenly, you can do your entire experiment on about 1/10 of a lane. The next stage in your analysis, once you have all of these sequences for all of those RAD tags, is to call variants. That is, for each RAD tag, I'm just showing you 3 here, but remember in our example, we have about 12,000. For each RAD tag, you will find the genotype for each individual that you have in your study. We have 200 fish in reality. Here, I'm just showing you 8 males and 8 females. So, sorry, four males and 4 females. So for these 8 total fish, I'm showing you their genotypes at these 3 different RAD tags. There are other sequences depicted by the white line here, but I'm just showing you a snip of interest that we found to be polymorphic in this population of fish. I am depicting the male fish with blue and the female fish with yellow. So as you can see here, here are the genotypes for each of the fish at this particular locus in each RAD tag. Now, to answer our biological question of can we find a genetic locus that segregates with sex, that is, that seems to be associated with sex, we just have to see that. Is there a genotype that associates with sex? Here, I don't see a particular association with ragtag 1. It seems like either males or females can be homozygous T, homozygous G, or heterozygous. Similarly, ragtag 3 seems to show no association, but ragtag 2, we see that every single male individual is heterozygous TC and every single female is homozygous C. And this is an actual summary of a study that was done. I can post the paper on Moodle. And in reality, it wasn't as simple as finding one locus that explained the male specific variation that they discovered, but they found about 250 male specific SNPs. And it did seem that most of the male SNPs were heterozygous, where the females were homozygous, suggesting that males were the heterochromatic sex in this species of tuna. But I'm going to stop here. This has gotten really long, but hopefully I've shown to you that you can reduce the genome size, only sequence some bits, and still be able to capture lots of variation that you can use to explore a biological phenomena that's interesting to you.